So good evening. Today is my week 7 problem solving session. So in this session, we'll be talking about week 7 topics, which were about large scale data science, such as CPTAC, NCI Cancer Research Data Commons, NCI Proteomics Data Commons, and DIA SWATHAtlas, SRMAtlas, and PeptideAtlas. So these were some of the topics which were covered in week 7. So we'll go through them one by one. And we'll also look into the large scale data science and what does it mean and how the data is stored and how it is managed. Along with this, we'll look into few MCQs and current literature. And if you have any doubts regarding the assignment or anything, so we'll take that also. So before beginning, I'll just confirm. So is it visible? Okay, thank you. Okay, so now let's start with the large scale data science. So what does large scale data science means? It means that when you are analyzing and processing massive data sets, which are otherwise not possible to analyze by using traditional data processing tools or methods. So that is known as large scale data science. And in large scale data science, you generally use advanced techniques, algorithms, and powerful computational resources such as cloud services or distributed computational frameworks or specialized hardwares. So that is known as large scale data science. And there are few aspects of large scale data science. There are few steps which you follow. So first is data acquisition. So in large scale data science, first thing is you need a data. So from where you can acquire that data. So high throughput techniques generate vast data sets by identifying and quantifying proteins in samples. So in this case, we will be focusing on the proteomics dataset, which is coming from mass spectrometry. But if you have to generalize it, then you can say the data can come from anywhere. But in this, we'll be talking in perspective of proteomics dataset. So high throughput data set which is generated by mass spectrometry techniques or liquid chromatography or omics integration and various data repositories. So these are the sources from where the data can be acquired. And then you can perform its large scale data analysis. Then comes data storage and management. So now if you have the data, you also need some place to store that data and manage it. So storage uh, includes cloud storage such as Amazon Web Services, Google Cloud Storage Space, and Microsoft also provides you the cloud storage. So these are few of the services which are provided for storing your large scale data set. And along with storing, you can also share it and you can also perform various analysis. So then third step is data processing. So now you have acquired the data, you have stored it somewhere. Now the third step is that you want to process that data because ultimate aim for that large scale data is to get some results from it. So we need to process that data. So processing of complex data sets. So why we does the complex data set processing? When we want to identify some proteins or we want to quantify certain proteins which are coming from mass spectrometry data analysis and modification analysis, if we want to check that are there certain modifications, post-translation modifications present in the protein. So all those things comes under data processing. And for processing, you can use distributed computing services or parallel processing, machine learning. So these are few of the techniques which you use for data processing. So last time also we looked into machine learning methods, that how machine learning helps you to analyze the big data or large scale data. Then next thing is after processing everything, after generating results, now the main thing is to analyze that result and interpret that what an analysis is giving you. So analyzing data to extract meaningful biological insights such as protein interactions and its functions. So for those interpretations, you can use software such as MaxQuant, OpenMS, Skyline. So in the last supplementary lecture, you have looked into Skyline, that how to use Skyline and how you can interpret your data set when you have different peptides coming from different mass spec experiments. So how you can analyze it and what you can interpret from it. For example, if you are comparing between normal person and a diseased person, then you can compare the expression of different peptides, you can quantify proteins, and different things can be, means you can plot that data to interpret the results. So you can also use heat maps or network analysis. So last time we looked into the volcano plots, that how volcano plots can help you to look into the genes which are overexpressed or which are underexpressed. So in that way, you can interpret a result visually. Then comes cloud computing and HPC. So for example, if you have a very large data set, then you require cloud computing or high performance computing in which your own traditional machine is not sufficient. So in that case, you can go for the cloud platforms so that you can perform your analysis on those uh, bigger machines, and you can also share that data. So this is an additional step, although it was also covered in data storage and management, but this is also an additional step. If you need to store it in the larger set, or if your data is so intense that its computation is not possible on the smaller machines, then you can go for the HPC, which is high performance computing, where multiple computers are connected together. So in that, you can perform your analysis quickly within fraction of minutes or seconds. Now we will look into some of the examples of tools, databases, and platforms which are generally used in proteomics research. So this is also about entirely about the proteomics. So when we talk about data repositories, so there are few examples of data repositories from where you can access mass spectrometry data. So one such repository is ProteomeXchange Consortium. So now there are multiple repositories and some are consortiums. So consortiums are the places where multiple repository data set you can access from one place. So consortium of proteomic repositories, it means it provide you access to mass spectrometry data coming from PRIDE, Massive, PeptideAtlas, JPost, iProx, and Panorama Public. So there are 6 different repositories which contains individually the mass spectrometry data, and these are hosted by different countries and different labs. So what ProteomeXchange does, it collects its data from multiple such repositories and provides you access at one place. So you don't have to go through all these one by one. You can simply go to ProteomeXchange and get your mass spectrometry data. So in the later slides, we'll see that how the data is arranged in ProteomeXchange and how you can see that how the data set is coming from different repositories. Then comes PRIDE. So PRIDE stands for Proteomics Identifications Database. So it is also a major repository which provides you proteomics dataset, which is also coming from mass spectrometry based experiments, but it also stores the information of protein and peptide identifications. So it will not only give you the peptide spectra information, but it will also give you the sequences. It has also identified the peptides, that to which protein that particular peptide belongs to, and what is the sequence of that peptide. Then the third example is MASSIVE, which stands for Mass Spectrometry Interactive Virtual Environment. So this is also a repository which allows you to share your data, disseminate your data, and analyze mass spectrometry data, which is hosted by the Center for Computational Mass Spectrometry at UC San Diego. So there are different labs which host these datasets. So similarly, this is hosted by UC San Diego, and the same thing it also does. It also stores mass spectrometry data, but along with that, it also allows you to analyze that data set. Then comes PeptideAtlas. So this is also a database which aggregates and organizes identified peptides which are coming from public proteomic datasets. So this is also a kind of a comprehensive map. So whenever different experiments are performed, so it collects that all the experimental datasets and store their information in it. Then comes SRMAtlas. So as the name suggests, it is specific for the selected reaction monitoring. So all the experiments which are based on SRM or MRM, which are multiple reaction monitoring, so only those experimental mass spectrometry data is stored in these repositories. Then comes SWATHAtlas. So SWATHAtlas is also like SRMAtlas. In this also, the data will be specific to DIA, which means data independent acquisition. So all those experiments where the data is generated with the help of sequential window acquisition of all theoretical mass spectra, that will be stored in this repository. So in the later slides, we'll also discuss about SWATHAtlas, that how it stores the data and how it perform analysis and how it is different from SRMAtlas and old shotgun sequencing techniques. So, okay, the next thing is data analysis tools. So there are other repositories also. In the next slides, we'll also look into that, but this is just an example. Then comes data analysis tools. So in mass spectrometry, the tools which you use are MaxQuant. So this is a software platform from where you can analyze your mass spectrometry based data set. You can perform protein identifications, quantifications, and you can also apply certain statistical analysis so that if you have certain hypothesis, so you can prove that hypothesis and you can accept it by performing statistical analysis so that you can be sure that your results are, means you're confident enough to comment something. Then comes OpenMS. So it is an open source software framework for analysis of mass spectrometry data, and it also supports wide range of proteomic workflows. So as we discussed in the previous class, that there are some organized sequential steps which are known as workflow. So OpenMS is like open source, anybody can access it. So in this also, there are multiple workflows. You can perform any kind of analysis on the OpenMS for mass spectrometry data. Similarly, Skyline is also a tool which is especially for the targeted proteomics, and it also allows you to create your method, such as setting up methods. It also help you to acquire the data from different machines, or you can also input your own data. It helps you to visualize that data and analyze the mass spectrometry data. And we can also plot the data after analyzing so that you can visually inspect and interpret that what are the results. Similarly, Protein Pilot is also another software for protein identification and quantification, and it also provides you with the advanced algorithms for data analysis, especially for the mass spectrometric data analysis. Then some machine learning tools are also available. So previously we talked about softwares. Now these are machine learning tools such as DeepNovo, which is based on machine learning. And in this, you can perform de novo peptide sequencing from mass spectrometry data. So there are certain cases where you do not have, for example, you have identified some novel peptides, and for that you do not have already available protein sequence in your dataset. So in such scenarios, you have to de novo create, you have to customize the protein sequences. So in such scenarios, you can go for de novo. So this machine learning tool will help you to create peptides by de novo peptide sequencing. So in this, you can also identify the novel peptides, whether they will be present in means to which protein they belongs. So in that case, you can use de novo. Then comes MS2PIP, which stands for MS peak intensity prediction. So this is also a machine learning tool which can help you to predict the peak intensities which are present in the tandem mass spectrometry. And these peak intensities will ultimately help you to quantify that in which range the particular peak is present, so that you can quantify the abundance of the protein or peptide in that particular sample. Then comes visualization tools. So after analysis, you can also visualize your results. So I have taken example of Cytoscape and Perseus, although there are other visualization tools and string and means other tools are also available. In Skyline also you can visualize it. So some of the visualization tool are Cytoscape. So Cytoscape is especially when you want to analyze certain networks. So in case of proteomic dataset, when you are looking into protein-protein interactions, so in such scenarios, if you look at their networks, that how those proteins are connected, so it will be more insightful. So in such scenarios, you can use Cytoscape. It will help you to visualize complex protein interaction networks and pathways which are derived from proteomic data. So I hope you're aware about the term network. So what happened here is that you have multiple proteins which will be shown in the form of either circles or rectangles, and those proteins which interact with one another, they will have some connection between them, which is referred to as edge. So in this case, in Cytoscape, you will have all the proteins, all the circles which are referred to as nodes, and if they are interacting with one another, then they will have some connection. So in this way, there are possibilities where one protein can interact with multiple other proteins. So this can give you the information that which proteins are most important, or which protein is the key protein in the particular pathway which is interacting with multiple other proteins. And if that protein will be either disturbed, or it will be if it will be regulated, then it can impact multiple pathways. So in such scenarios, Cytoscape will provide you the visual, means you can visually see that. Otherwise, if data is arranged in the table, then you won't be able to analyze it. So Cytoscape is like visually you can see it in the form of network, so that you can quickly analyze and interpret that which of the proteins have more number of connections with other proteins and which is having lesser connections. So in that case, that protein might not be important in that particular pathway. Then comes Perseus. So this is also a platform for statistical analysis and visualization of quantitative proteomics data. And it is sometimes integrated with the MaxQuant. So whenever quantification is done, so it is integrated with MaxQuant, and after quantification, you can use Perseus to visualize the results. And these results can be visualized in any form like heat maps or plots. Then comes data integration platforms. So you can also integrate your data. For example, after performing mass spectrometry, you have also got the information about the quantifications. Now you want to see that to which other proteins this particular protein is interacting. For example, in cancer, I have obtained some P53, for example, I obtained P53 or some any other protein I have obtained. Now I want to check that in which pathway it is involved or which are the other proteins it interacts with. So I can go for string, which is known as search tool for the retrieval of interacting genes or proteins. So in such case, I can simply provide my data set in which all the proteins are present, and I can check in string database that which are the other proteins to which it interacts. So string is a database which contains the information of all the protein-protein interactions, all the experiments and computational predictions, and text mining is done. So it will take the help of all the available information and will return you the output. So in that way, you can check that whether the proteins which are present in your sample, either they are interacting with other genes or other proteins. So in this way, it can help you to generate some insights. For example, if your protein is interacting with many other proteins, so in that case you can identify something, or you can also know that this particular pathway my gene is involved. Then comes UniProt. So as string provides you the protein-protein interaction information, whereas UniProt will provide you entire detailed information about the protein. For example, from its structure, sequence, functions, and its gene ontologies, that in which particular cell component it is expressed, what is its function, what pathway it is involved in. So UniProt is also a kind of a big repository which provides you the in-depth information of a protein. So in such scenarios, you can integrate UniProt to your mass spectrometry data, and you can analyze that what functions this particular protein performs. Then there are cloud platforms where you can store your data, such as Amazon Web Services, Microsoft Azure, and Google Cloud. And similarly, in high performance computing, there are certain computer clusters which are already available, such as EMB and EBI cluster or XSeed. Other than this, there are other clusters that are also hosted by different institutes. So these are some of the examples, but there are other means clusters which are also available in other institutes, in which you have high performance computers which are connected to one another over internet. So you can access them. You can ask them to provide you access to these computers, so that if you have a larger data set, then you can perform your analysis on these systems instead of performing on your traditional computer system. Yeah, so now we'll talk about ProteomeXchange. So is everything clear till now? Okay, so now let's see what is ProteomeXchange. So as we have seen that it is a consortium which will collect the data from multiple different repositories. So it has 6 members: Pride, PeptideAtlas, Massive, JPost, Panorama Public, and iProx. So in this also they are providing you the different locations. So these all repositories contain mass spectrometry data. Along with that, each one of these repositories provide you some kind of benefit, such as some provides you to store the data, submit the data, or some provides you to analyze the data, visualize the data, or some allows you to input your data files, process data files. So in that way, these are different repositories, and all these repositories combinedly are stored in the ProteomeXchange, or you can say ProteomeXchange is a common point from where you can access the data present in different repositories at one place. So you do not have to individually go to each of them. You can simply go to ProteomeXchange, and this contains raw data, IDs, and quantification data as well as metadata. So these types of data can be accessed from ProteomeXchange. So there is an option called as access data. So if you want to access the data which is stored in the ProteomeXchange, you can simply click on access data. If you have some data related to mass spectrometry experiments or SRM data, so you can submit that data as well. So ProteomeXchange provides you the means flexibility to exchange the data. You can also get the data and submit the data as well. And you can also subscribe. For example, different repositories, each year the data gets updated, new data gets added, or some data gets changed. So you can subscribe to it so that if any new announcement comes up, so you can get the information. So now if you want to access the data, so we'll simply click on the access data, and it will take us to this particular home page. So now first thing which you should see is that how many number of species data it contains. So in ProteomeXchange, there are top 10 species for which the maximum amount of mass spectrometry data is available, although it contains the information of other species as well, but data related to them is quite less. So in this, it provides you the list of top 10 species for which maximum data is available. So this is the current information. So currently there is 47% mass spectrometry data which is coming from Homo sapiens. So there are 17,288 data sets which are present in ProteomeXchange, and these data sets are taken from 6 different repositories. Then top 10 instruments. So as we know that in mass spectrometry there are different instruments which are generally used. So this is the list of top 10 instruments, and the maximum 12% is contributed by means from where 4315 datasets are coming is coming from Q Executive, Hive, HF, and then top 10 keywords. So keywords are also the words which you search, and there will be multiple datasets which appears. So similarly, top 10 keyword information also can be seen here, such as in this case, LCMS is the most searched keyword. And then in USI, it is the if you need to access the individual spectra from a ProteomeXchange dataset, then you can click here. Now, if you look at the data set, so if you want to specifically look into some particular species data set, then you can simply go to top species and click on the species. So in that case, you will only be shown the data set related to Homo sapiens. But in case you generally want to see that what are the data sets present, then you can simply scroll it here. So this first thing is providing you the dataset identifier. So in ProteomeXchange, the mass spectrometry data, which is coming from multiple repositories, it will be given some identifier or some ID, which will uniquely identify that particular dataset. So PXD stands for ProteomeXchange dataset, and then there will be a number which will be given depending on the time, means at what sequence level it is coming. So so there will be a title. So means because different experiments have been performed, so there will be some title of those experiments. So this will be the title. Then it will mention the repository because it has 6 repositories. So it will also tell you that from which repository that dataset is coming. Then it will mention the species, that for which species that experiment was performed. So first experiment was performed for mus musculus, which is the mouse, and then it will tell you the instrument which was used. So Orbitrap was used and Orbitrap Fusion was used. Then it will also tell you about the publications. So there are, so one more thing which I want to add, that in ProteomeXchange, it also means you can submit your data which you have already published, or if it is not published, then also you can submit your mass spectrometry data, and then they will analyze it and they will check that whether it's accurate. So in this, it's not that always the published data is submitted, but sometimes the publishing is pending also, then also you can submit the data. So if your data is published, then it will also provide the link from where you can access the publication, that what was actually done in this experiment. So this is just the data set, but in by reading their paper, you can also see different things, that how they've interpreted their results. And then it will also tell you the lab head, that who was the head of this particular data set, who has designed this data, and who is the lab head, and announce date when this data was announced. So this was all which you can find here. And similarly, if you scroll down in this, so you can find the announced years. So each year new data sets come up. So it will tell you that how many new data sets are added each year. So in 2024, 5,337 new datasets have been added. Then it will also tell you the top instruments. So this is the list of all the instruments which are generally used by these repositories when they were collecting the dataset. So these were the tools which were majorly used, instruments. Then top keywords. So because each of these experiments or each of these publications, they will also provide you some keywords so that you do not have to write the entire title or entire topic. So you can simply write a smaller keyword. For example, if you want to see the liquid chromatography mass spectrometry data, so you will simply write LCMS instead of writing the complete title. So in that way, by providing keyword for each of their experiments, you can quickly access their data. So that's why they keep some keywords with their data. So these are few of the keywords which are majorly searched, and these are the number of times, means number of data sets which contain these keywords. Then comes repositories. So maximum contribution of the mass spectrometry data is by PRIDE. It gives around 28,196 data sets. Then iProx, then Massive, JPost, Panorama, and PeptideAtlas provides 140 data sets to ProteomeXchange. So these are all things you can find when you will scroll down that homepage. Now next thing is data submission. So now you have accessed the data. So next thing is that if you want to submit your data, then you can simply click on submit data. So there are certain guidelines which you have to follow while submitting your data. You cannot directly submit anything. You need certain things before submitting your data. So when you click on submit data, then you will be taken to this home page in which it will tell you that what kind of submissions it supports. So it supports the experimental submissions which are coming from all proteomics data workflows. So there are different repositories where you can submit. So ProteomeXchange, you cannot directly submit to ProteomeXchange, but you can submit through Pride. So in Pride, it will take the GUI-based PX submission tools, or you can simply go to the Pride site and they will provide you the link from where you can submit your data. But before submitting any of your data, firstly, you need to create your ID. Then you can submit your raw mass spectrometry data or quantification data. So that is one thing which you have to do. Similarly, after Pride, PeptideAtlas, PASL. So this is also a means service through which you can upload your data or submit your data to the PeptideAtlas. Then Massive. So in Massive also you can submit your data. So in Massive, you generally provides shotgun proteomics data, whereas in PeptideAtlas, you provides the SRM and MRM data. So now we will look to the Pride that. So if you go to Pride, you can click on this link if you want to go to the Pride search engine. So in Pride, it is the proteomics identification database. This is the homepage of Pride, and it tells you about the what Pride archive stands for. So in Pride archive, it is a database which is a centralized and standard compliance public data repository which contains mass spectrometry data, proteomics data, and what information it contains is it contains the protein and peptide identification information. Along with that, it also provides you the expression values, post-translational modifications, and supporting mass spectra evidence, such as raw data and the peak list files. So all these informations are present in Pride Archive, and all the public datasets are available in Pride Archive in the repository, and it can be accessed by the website. So you can simply go to their website, or you can also use their API through which you can, from command line, you can access the entire data. Then the mission of Pride. So ultimately, the mission of all these repositories is to collect more and more data and allow easy sharing of that data. So that means if there will be only one data set and it is not interacting with the other repositories, so there will be a lack of data and lack of information. So in this way, to store your time and ease your experimentation or analysis, so there is a mission that each and every data repository shares their data, update their data, and provide information if there are some updates. And it tries to generate more and more mass spectrometry data so that later on, if we talk about in perspective of disease medicine, so if more and more data will be available, then better medications can be designed, better drugs can be designed, or better understanding can be generated related to proteins. So this is about the Pride, means information related to Pride. Now let's see that how we can submit the data. So this is main page which appears in Pride. So that was about of the Pride, in which every information was depicted. Now, if you want to see its home, so in this you can search for any data set. For example, if you want to search for a particular peptide, or if you want to search for a disease, so all these things you can enter and you can search for it in the Pride archive, or you can also use Pride Archive USI or Pride cross linking. So these are different means places from where you can access their data depending on what information you want to see, or you can also search the data. Now there are some means guidelines that how you can submit the data in Pride. So first thing is that submitted dataset property trees. So there are different categories in which the Pride contains the data. So it takes the data for Homo sapiens, Mus musculus, Ratus norvegicus, and Equus kibalius, which is horse. So the information which it contains is like about cell culture, epithelial cells, blood, erythrocytes. So these are different samples from where the information is given. So this is just a tree which tells you that what kind of data sets have been submitted to Pride earlier. So if you click on more, then it will give you the more larger table and all other species also from where the data has been acquired. And in this, it shows you number of submissions that with each passing year, more and more submissions are happening. And it is also telling you about different countries from where the more and more data has been submitted. So these circles represent different places from where the data is coming. Now let's look at the steps that how to submit your data in Pride. So first thing is you have to register. So firstly, if you register, then you will choose the submission type in which you will mention that what is your type of submission, what kind of data you have. Then you will submit your data set, and you can accessing private data. So in certain scenarios, some people keep some layers that who can access your data and who cannot access your data. For example, if my data is not published yet and I do not want my competitor group to access my data, so in that case, I can ask for some privacy so that means who can access it, or person with the particular password or some means I can decide that who can access my data, or I can simply keep my data entirely private till the time my publication is out. So these all things you have to decide while submitting the data. Then comes post submission, that after submission, if you have some updates or if you want to make some changes, so that comes under post submission, and all the processes which are followed after the submission, they will come under post submission. So you can download the submission tool in which it will tell you the step-by-step information. It will gives you the step-by-step steps, and you can follow those steps to submit your data. And you can also read their guidelines that what information you have to provide and what should be the confidence level. So because quantification data also you have to provide. So all those things you can see. If you want to read more about it, then you can click on this particular link. So PRIDE is hosted by EBI, which is European Bioinformatics Institute. So you can go to their site and you can read more about PRIDE, that what other information is available. And if you have any query, then you can also ask through their chatbot different questions. Now this, so the thing which we looked was at home of the PRIDE. Now if you want to see the resources, so resources are PRIDE Archive, Archive Annotated, Archive USI, and Archive, PRIDE Spectral Libraries. So these are different resources. Depends that which data you want to access. You want to access the spectral libraries, or you want to see the annotated data, or simply the mass spectrometry identification data. So for example, if we click on Peptide Archive, then it will provide us the archive, the list of data sets. So first, it gives me the ProteomeXchange ID, which is given for this particular data set, because these repositories are stored at the means it they also share information with ProteomeXchange. So in ProteomeXchange, they provides the unique IDs to each and every database in each and every data set. So similarly, it will have the unique ID coming from ProteomeXchange database. So this is the ID. Now it will tell you about the title. So all those things which you have seen in ProteomeXchange, similarly that thing is shown here, but in that case we have seen it in the form of tables, and in this case it is shown in different separate rows. So in this way, if you want to access that data, you can simply click here and you can access that data which is available. So PRIDE Archive will contain all the data sets which are coming from different experiments which are submitted to PRIDE especially. Then comes PESEL. So that was all about PRIDE, about how to submit the data and how you can access the data which is already available. Now PESEL is like PeptideAtlas SRM experiment library. So this is the part of PeptideAtlas only, but PeptideAtlas is not specific to SRM or MRM. So PESEL is specific to SRM experiment library. It is component of PeptideAtlas project, which is designated to enable submissions, dissemination, and reuse of SRM experimental results. So in this, you can browse the SRM experiments. You can also, if you have any query, for example, if you have any peptide sequence or anything, so that comes under query. You can simply ask for particular peptide sequence or protein sequence. You can ask this PASL and it will provide you the results which contains the information. So each and every data set will have some kind of peptides or protein information. So it will provide you with that information, that in which data set your query is present. You can also submit an SRM dataset, as we have seen that to submit the data, you can use the PASL. And you can also access the pre-publication data with the reviewer password. So that, which I mentioned previously also, that you decide that to whom you want to provide the access. So sometimes when you have not published your data, you are not confident enough and you do not want to share your data, then you can hide your data and you can provide some restrictions. So in such scenarios, what happens is that because the people who are reviewing your paper, so before publications, there are a group of peer reviewers who review, who check that whether your analysis is correct or not, whether it is confidential enough or not. So in that scenarios, what you can do is you can provide access to the reviewer only so that they can check your data set. So uh, pre-publication data with the reviewer password. So in this case, if you are a reviewer or if you are a person who can review that data, and if they have provided you with the password, you can simply write password here and you can view. So in such scenarios, you can also check, or you can also look for the data which is not published yet. Yeah, so if you go to PeptideAtlas, so this is the home page of PeptideAtlas. So previously we have seen PASL, which is specific to SRM dataset. Now PeptideAtlas is generalized. It is not specific to SRM or MRM. It is for all the mass spectrometry data analysis. So in PeptideAtlas, it is for multi-organisms. So other than human, it also contains other organism information as well. And for human also, each and every year, whenever new information is added, it keeps on updating, it's built. So each and every version is referred to as build. So in this case, now currently human all 2024 build. So it will contain all the information related to human, which is coming from different mass spectrometry experiments, which are submitted to PeptideAtlas, and means in 2024 it will be updated. So it will contain information, latest information also and previous information also. So if you want to access all the mass spectrometry data, so you can simply click here. Similarly, other than for entire human, you can also be specific for the plasma. For example, if you are interested only in the plasma proteins, so in that case you can simply click on human plasma. Similarly, human plasma, extracellular vesicle, or human urine. So similarly, there will be different human builds. There will be coming from different samples. So in PeptideAtlas, depending on different experiments, you can access the mass spectrometry data. So along with the spectral information, it will also provide you with the sequence level informations, quantification informations. So all those things can be obtained from PeptideAtlas. So in the left side, you will see that there are different means in the menu bar, you can see different options you can access. So this is the home. You can also read their overview or context, publications, softwares, database schema, feedback, funding, frequently asked questions. So this is just a generalized view. Then comes Atlas data. So what kind of information is stored? So if you want to access that, you can simply click on data repository. It will tell you the entire information. It will give you the information of all the data sets which for which the information is available in PeptideAtlas. Similarly, HPPP Data Central, which is specific to human plasma protein information. So this will be specific to human. Then PeptideAtlas builds. So it will tell you all the builds. So as I mentioned earlier, that builds are like new versions. So whenever new version come up, it is referred to as build in PeptideAtlas. Similarly, for all the organisms for which it contains information, you can access multiple builds, which will be the updated mass spectrometry informations. Then PeptideAtlas exports. So you can also export their data if you want to download it. Then there is THISP. Similarly, it is also for the Homo sapiens data. And then search databases. So you can click on search database and then you can search this particular database. So databases like data repository, which is PeptideAtlas, so you can search it. You can also contribute data if you want to submit your data. So you can click on contribute data. And it also provides you link to the related repositories such as SRMAtlas, PASL, SWATHAtlas. So means this also contains mass spectrometry data specific to SRM means SRM experiments. PASL is also for SRM, and similarly SWATHAtlas is for DIA data independent acquisition experiments. So these are the related links that will take you to these particular repositories. Then spectral libraries. So if you want to access the library information or spectra ST search, so you can click here. These all are the informations which are available in PeptideAtlas, which you can easily means access. Now, if you will scroll down, then you'll find there are different tools and data. So if you want to access raw data repository, then you can simply go to raw data repository, or you can go for this, which is the tired human integrated search proteome. So in this also they have organized the available mass spectrometry data related to human, and they have organized it into different proteomes and arranged it in particular order. Then proteomapper, in which you can map the proteins or the means match those peptides to entire proteome. At protein level information it will contain. Then comes PeptideAtlas builds, which will provide you the information of all the available builds. So there are some people or there are certain papers which have performed experiment on certain build. For example, some paper came up in 2012. So if I want to check that whether how they have performed analysis, so in that case I cannot proceed with the current build. So if I want to make sure that how their workflow is working fine or not, so I have to work with the build which they have used. So in such scenarios, this peptide atlas build is helpful because it will provide you information of all those builds which were previously available. So in that way, you can access the previous build also and check their workflow and check that whether similar results are also reproduced or not. So in such scenarios, it will tell you that whether their analysis or their workflow is working fine or not. So in such scenarios, you can use peptide atlas builds. Then you can also explore builds, proteins, peptides. So these are few options to browse. And you can also query. So what kind of query you can give? You can give proteins or peptide information, and you can check for the information available in the dataset, which is present in Peptide Atlas. So you can click on download if you want to download any information, or if you want to search something, you can simply click here. So to means read more about it, you can click on this link. It will take you to Peptide Atlas. So you can scroll and check that how to work with Peptide Atlas. So if you click on raw data repository, so it will provide you this kind of information. So as we have seen in ProteomeXchange, similarly, this peptide atlas also provide you the information in the form of a table. You can download also, you can download this list. You can download it in the form of a table. And if you want more information, then you can simply click on these plus signs or in this particular accession number. So it will take you to that particular experiment, that particular data set. If you scroll down, then it will contain more and more detailed information about the experiment. So first thing is the accession number. So accession number is also like a unique ID. So as in case of Pride, we have seen that it contains the ID of ProteomeXchange only. But in case of Peptide Atlas, the ID is of Peptide Atlas ID. So this is PAE, and then it will tell you the sample tag. So that it is for the tagged dataset. So tagged mass spectrometry dataset. So what tag was used? So that information. Then sample title. So what is the title of the experiment? For which organism this experiment was performed? Which instrument was used? And what is the publication? So other than this, there are other column information are also available. So you can search for those columns, and in here in this you can, if you click here, then it will also provide you other information as well. So in that way you can decide which columns you want to see. And if you click on this accession, this will organize your accession numbers in a linear order, in ascending or descending order depending on you. So this is just to sort these values. So this is all the information related to the raw data, and you can simply download them. And in the left side, similar thing is shown here. What are the different informations you want to download? Yeah, so this is all about the peptide atlas, that how you can download different information and how to access the information available at peptide atlas. Similarly, the third repository is massive, which is also providing the data to ProteomeXchange. So this is hosted by Center for Computational Mass Spectrometry. Massive stands for Mass Spectrometry Integrative Virtual Environment. So, interactive virtual environment, and it is a member of ProteomeXchange consortium. So in the home page, it provides you the entire information. So statistical information about this repository. Firstly, it tells you about the public data sets, that how many data sets are available. So 15,825 data sets are available. The number of files. Then what is the size of this particular repository, which is 635.55 terabytes. Then spectra. So how many spectral information is available? How many people have subscribed to it? How many protein information is available? How many peptides are present? Peptide variants, PSMs, modifications. So these all informations are given in homepage, and you can search their dataset identifier or metadata. So similar to Peptide Atlas and Pride, they also have some identifier. So if you have the information of that identifier, then you can simply search that identifier. In case you do not have that information, so you can also use the metadata information or universal spectrum identifier. So for each spectrum, there will be identifier which is unique. So in these two search bars, you have to have the information of the identifier, then only you can search. But if you want to means overview that particular repository, then you can simply go to access public datasets, and you can simply access the available datasets which are present at Massive. You can also submit your data, and which will be shared among the all the means people in the ProteomeXchange. So you can submit your data, you can search identifications, and you can reanalyze the spectra. For example, you already have a spectra, so you can also reanalyze it. Or if Means, for example, in massive also, you can download their spectra and you can reanalyze that massive also. So in such scenarios, you can use massive for that. You can reanalyze the public datasets which are already available in this. So for that particular analysis, you can use MSGF plus database search or split spectral library search or MODA. So these are some of the example of tools or softwares which you can use for the reanalyzing spectral information which is already available in the public datasets. If you further scroll down, then you can also see that you can compare the results. So result comparisons, you can share the reanalysis. For example, for particular spectra raw data, you are performing analysis. So you can also share that analysis if it is novel, if it is different from already performed analysis, because each and every person have a different way to analyze the data or interpret the data. So in that cases, if your analysis is novel or it's new, which was not previously done, if you have some new pipeline or workflow, so you can share your reanalysis for the already available data. Then comes Protein Explorer. So in this, you can explore different proteins for which the information is available at Massive. In massive.quant, you can search for the quantification of different peptides coming from different data sets. So quantification data can be accessed. Then Massive Knowledge Base. So this is like a big community, a complete like encyclopedia. It is a knowledge base which contains all the informations which are related to mass spec data. So you can go for the massive knowledge base if you want to search all the data which is available. Then comes Corona Mass KB. So this is also a knowledge base which contains the especially the data which is specific for the coronavirus. So in such scenarios, you can go to Corona Mass KB, which will contain the coronavirus specific mass spectrometry data. So this is about the current homepage of Massive, that what are the informations which you can access from Massive. Then if you look at the Massive data sets, then these are the data sets which are also available in the form of a table. So in Massive, it has a ID which will start with MSV. So this is the ID, and it also provides you the ProteomeXchange ID as well. So this is the Massive ID and this is the ProteomeXchange ID. And it also provides you the title and what is the submission type. So this tells you about what is the submission type, whether it is a partial or a full submission. So in that case, this is the status, and then this is the entire description. If you want to read in more detail that what was exactly done in the experiment, you can go for the description. It will tell you entire detail that what are the different methods which were used for data acquisition, or means what are the tools which are used. So whether it was DDA, DIA, then there are keywords. So each means each data set has some keyword through which you can search it. So in this case also it has the keyword Q executive. Similarly, principal investigator. So the person who has submitted it, who was the principal investigator, that name is given. Because this name was given because if you want to directly contact that person, if you reanalyze their data or if you have any query related to their data, then you can simply get the information of principal investigator. You can directly contact them if you need any help, if you want to understand anything, so you can contact them. Then species, which is mus musculus, then instrument which was used, then post-translational modification information. So if the data set contains post-translational modification, then it will tell you information about it. Then uploaded by, so who has uploaded it, from which system was it uploaded? So from CCMS system, and the upload date. So it will also tell you the date when this particular data was uploaded. So this all information is present in the massive dataset. And other than these, other columns are also available. So from select column option, you can look for other column information. So for example, in certain cases, we might not need the information related to upload date. So in that case, you can means discard this column, and there are certain other columns which might be beneficial. So from select columns, you can check that which columns are of your interest. So in that way, you can turn on those columns and you can simply export these results by clicking on export filtered results. So yeah, and if you want to take the data from a single particular data set, so in that case, you can simply click on this MSV. So for this particular experiment, it will provide you the data set. Because when you export these results, so the exported result will be in the form of a table. It will not be a data set. So to access the dataset, you have to click on these individual identifiers, or you can simply copy it and search it on the search option, which we have seen in the previous slide. So this is all about the massive dataset. And you can also perform reanalysis. So as we have seen in the front page that there are different tools. So you can simply download their data set and go to reanalysis and then check that you can analyze the spectra. You can identify the peptides. You can compare them. You can decide that which peptides to compare. So that comes under reanalysis. Then journal information, UCSG, proteomics, future tools, demo, contact. So these are some of the means menus options which you can scroll if you want to read more about this. Now let's move to fourth repository which provides data to ProteomeXchange, which is iProx. So let me just ask, uh, are you following? Till now, is it clear? Okay. Okay, so ProteomeXchange is like a common place from where you can access different data, means different data present in different repositories. So ProteomeXchange is not like a repository, it is like a means combined place from where you can access them. So it is like a single point access thing. So to submit the data, you have to submit into different repositories and then they will share their data, and in ProteomeXchange, you can simply access it. So for uploading, for submitting, you have to go to these repositories such as Pride or PeptideAtlas or PASL or Massive. So you have, and then what you have to do is that if you are submitting it, so these all repositories are connected in ProteomeXchange. So you do not have to submit it into different repositories. You simply submit it into one single repository. And it also depends that what data you have, because each repository have some guidelines that what kind of experimental data you can submit. So in that case, you can decide, for example, if you have SRM dataset, so in that way you can specifically submit it to, for example, PASL, or if you have data independent acquisition and SWAT mass spectrometry data, so you can simply submit it to SWATHAtlas. So similarly, it depends that what data you have. So each repository have their some standards. So in that case, you have to submit data to a single repository, then that particular data will get some ID, because whenever you provide your data, you have to generate your means ID. So in that case, you will have some ID, and then after submitting your data, they will provide you their ID. So for each particular repository, you will be provided ID specific to that repository. Now that repository will share your data to ProteomeXchange. So ProteomeXchange will also provide you ID, which will be unique. So yeah, and yeah, so you can submit the data before publication also, and it depends that when you want to submit it. So if you want to, because sometimes what happens is that we have different collaborators in different places, and in that case, we cannot share the data on mails or something. So these are the kind of platforms where you can share your data, because you can submit the data and you can decide to whom you want to provide the access. So in that case, many people, what they do is that they have some data set, they simply upload their data here, because these repositories, they are not just taking your data. Sometimes they also provide you the advantage to analyze the data as well. So in that case, you can simply submit your data and you can use their tools to analyze your data. Because if you have mass spectrometry data, you can also use their tools to get the information of the sequence, or you can visually see your data. So in such scenarios, even if you haven't done publication, you can submit your data and you can decide who can access your data. So in that sense, you can submit the data. And there is no such need that you always, it's on you. If you want to submit, you can submit. Otherwise, if you want to perform analysis on your particular system, then you can do that also. But yeah, this is one of the advantage that if you submit here, then anybody can access it. And there are strict guidelines through which your data will be protected also. So that is it about this. So when we say raw data and metadata, so metadata is like data about data, and raw data is the normal raw data, which is not completely processed. So in such scenarios, metadata, even if you don't have metadata, you can still proceed with the submissions. But the basic thing which you require is the raw data, which you have to submit, and ID also, you need the ID. So you have to generate the ID, then raw data, and even in certain cases, quantification data is also required, because raw data should have some kind of quantification which has done. And metadata is like additional, you can say addition, add on to that. If you have metadata, then you can submit. Otherwise, generally, when we submit our data, then after performing all the analysis, then metadata is generated. So while submission, we do not require metadata, but if you have, you can submit it. But otherwise, basic requirement is just the raw data. You need the ID, you need the quantification data, and it varies from each repository that what information they ask you for, because some might ask you for more information or some just need the raw data. Yeah. And this repository ID or your particular account that will be provided you by those repositories. So you do not generate it, but it is generated by them. And each dataset will have only single unique repository ID. Even if you submit your data again, because if your submission is partial and if there is some error, I mean, if you're resubmitting your data or you're updating it, so in that case, you will get the new version of your data. So ID will remain same. There will be a add-on to the version. So in that case, it makes sure that your ID is maintained. So you are not allowed to submit data again and again on different repositories. One submission is enough. Okay, so now let's look into iProx then. Okay, so iProx stands for Integrated Proteome Resources. So it is also Integrated Proteome Resource Center and it is present in China. So this is the repository which is present in China, which means these people host it. So it also contains the information related to mass spectrometry data, and it has 5,366 projects coming from, means 4,042 are coming from public projects, and these are the sub projects data files. So in the basically in the home page, it provides you the entire information, statistical information about a particular repository, that how many data files are available, what are the data sets, for which organism the data is available. So in this case also they are providing you this particular statistical information, and means how much data is generated last week, how many data files were added, what was their space, storage space, then in last month, last year. This is just for comparative analysis, that how your repository is performing, whether new data is being added or not, whether this repository is credible or not. So for that purposes, this information is shown. Then you can also submit your data to iProxX. So in the home page only, they provide you the link. So you can simply click on submit data by web. So you can online submit, upload your files to the iProx, and it encourages and welcomes the users to submit their proteome experiment, raw data and metadata. So as we discussed, that means metadata is not always required, but certain repositories, they might ask you that if you have a metadata, then you have to submit it. Raw data will always be needed by each and every repository. So for example, in iProx, they also ask you for the raw data as well as metadata. And along with this, they also ask you about the protein and peptide identifications, and which is to be published in peer-reviewed publications. So for example, if you want to do some publication, so before that publication, if you want to go for the peer review, so in that cases, you can submit your data. But this is not the means you can say this is not the basic requirement or primary requirement. They are asking for the metadata in scenarios where you want to publish it. So in this case, you have to provide the metadata. But what other repository does in such cases, because before publication, there are certain scenarios when you only have the data and you have not performed its analysis, you do not have metadata yet. So in such cases, what you can do is you can simply provide the metadata, and with time you can add on more data. So in such cases, they will mention that your data set is partial. So as we have seen that there are some submissions which are marked as partial. So that was the case where the entire information is not provided to this particular repository. So you can add on the information as you proceed with the experimentation. So similarly in this, you have to provide the metadata, along with that protein and peptide identifications, and which is to be published. So means before publication, you can provide it to iProx. Now this, if you look at the statistics, so this will tell you about the top 10 species which are majorly present in the iProx. So majorly Homo sapiens is present, because we know that most of the experiments are performed on Homo sapiens, means Homo sapien proteins, because these are easy to access, the and samples are easy to access, and this is also most importantly required. That's why most data is available related to Homo sapiens, then to other species, then comes others and mus musculus. So for human, there are 1,724 datasets which are available in iProx. And total data sets which are available are 4042. And this is just a diagram which tells you that how many for means which are the top 10 species for which maximum data is present. So Homo sapiens is dominating, and then comes JPost, which is the fifth repository, comes under ProteomeXchange. So JPost is Japan Proteome Standard Repository. So this is hosted in Japan. So these all repositories are coming from different countries. And why we are kind of using consortiums or means collaborations, because each country have different environment, different means different people have different protein expressions. So there are some epigenetic factors. So in such cases, their proteome expression or their proteins might vary. So in such scenarios, if we are taking data from different countries, so it will be a better way to get more and more data so that personalized medicines or better understanding can be done. For example, if USA decides and all the labs are coming from USA only and they are sharing data, then that would be more helpful. But in such cases, in consortiums, what they does is labs are from different part of the countries and different countries or different continents. So in that case, it elaborates or it enhances the data set. That's why they have taken. So JPost is the repository which is established in Japan, and it stands for Japan Proteome Standard Repository or Database. So this is the homepage of JPost. From repository, you can access the raw and processed data, which is related to mass-based proteomics. In the database, you can also means reanalyze their results with the unified criteria, and this will provide you information of all the data sets which are available in JPost, and from those databases you can download it and you can perform the reanalysis. So it also provides you the workflows. So this is also the advantage, because if you have not published your data, because before publishing you need some results. So for analyzing also or for designing the workflows, you can simply submit your data and then use some workflow to analyze your data, or you can use means already available data also you can reanalyze it and publish it. So in such cases, you can reanalyze the already available data and then you can upload it here. You can means upload the reanalysis results and you can also publish them on the paper. So that is the advantage of these repositories. Then from workflow, this will provide you the reanalysis workflow, which is used in JPost. Then for COVID-19, then help, gadgets. So it provides you the collection of tools which they have developed, which are specific for mass spectrometry based proteomics. So in that case, you can analyze your data, or you can analyze the data which is already present. Or if you have data, then you can use these tools to analyze your data. So now let's look at the database. So when you click on database, it will tell you the information which is available in JPost database. So first thing is it is telling you about the datasets which are specific to Homo sapiens. So there are 514 datasets which are specific to Homo sapiens. So there are 89.1% of all the datasets are for Homo sapiens. So JPost is not that much big repository. It contains lesser number of datasets only. I think total 577 datasets are available and 31,493 protein information is available, and there are 522 projects. And this tells you about the organs and diseases. So different diseases are present and their percentage. So if you place your cursor on these particular pie charts or this particular place, then it will tell you which disease is mostly studied. So samples are taken from which particular disease. So in that case, this is the information, and 514 human data sets are present in JPost. If you want to search something, then you can write the name of the species or the organ. For example, if you want to study ovarian cancer, or normally the the proteins which are expressed in the, for example, ovaries or lungs, liver. So in that case, you can also write the name of that particular organ, or you can also write the name of particular disease if you want to search for the particular disease specific peptides. So these are different filters which you can write and perform your search. And if you will scroll down, then you will find that they will provide you the project ID. So as we have seen that each repository has some unique ID. So in this case also that it provides you the project ID, which will be the JPost database. So it starts with the JPST and then the number followed by the number depending on the serial number, and then project title, species on which the experimentation was performed, and then comes data sets. So how many data sets are available? For example, if I am performing experiment on Homo sapiens and my uh, there are two samples on which I'm working. So there will be two different data sets. So in such cases, number of data sets can vary. For example, if I work on multiple organisms and I'm checking among them, so in that case, I will have more and more number of data sets. So they will mention the number of data sets which are specific to one single project. That's why we have only 522 projects, but data sets are 577. So that is the case that means one project can have multiple data sets as well. So similarly, there are only these four columns, these four means fields for which information they provide. Rest other information is not provided by JPost, such as which tool was used or other things. But if you want to read in more detail, then you can simply click on the project ID and then it will tell you the elaborate information about how the data was collected in this particular project. And you can download that data set also. Now, this is the workflow that means how the submission and how the data is means shown in the JPost. So this is the workflow that after post raw data. So you have the raw data and you want to means you have done it. You have done its analysis, quantification, and all those things. Now you will provide it to the repository. Now what happened? First step is that after processing the raw data, you will provide it to the repository. Now, repository will take your data and it will provide you with the ProteomeXchange ID. And along with ProteomeXchange ID, firstly they will generate JPost ID. Then your data will be shared at ProteomeXchange. So it will also generate ProteomeXchange ID. So that ID will be generated. Your data will be registered in a repository, which is the initial data set for JPost provided by JPost member. Because if you are a member of this repository, then only you can submit. So first thing that you have to be the member. Then they will register your data in JPost repository. Then they will reprocess it and results will be taken. So not they, but if you want to reprocess it, then you can also perform its reprocessing because we are submitting our raw data and JPost is also providing us with different analysis tools or different other things. So after submission, we can also go for reprocessing. Then results will be generated. Those results will be tagged into database and it will be arranged in the form of a cube. So these are the computational terms. So then these cubes will be aggregated. So cubes are nothing but these are the small data packets because your database you will have multiple different information sets. Multiple files will be generated. So these are like a different files which are present. So it will aggregate all the files and in the globe means it's a common place where all those files related to your particular data set will be kept and then it will search and it will provide means it will keep it in the form of user interface. So for example, as we have seen that it is arranged in the form of columns and it is arranged in the form of a table where different columns have different information about the data set. So your data will also be arranged in that form so that you can simply click on different columns what information you want to take, so that will come under user interface and ultimately whenever you will access it, so you can view it in the form of different either plots or pie charts or trends. So this comes under the database. So this is the entire workflow that how the data is submitted and how the in the end user can view it. You can either keep it for yourself or you can provide it means you can public your data so that everybody can see. So these are the results which are generated after analysis. But along with this, you will also have the dataset information which will be arranged in the slices. So it depends that what user want to see. So in that way, even if somebody only want to see your results, so they can simply click on your result portion. If they want to see the dataset, then from these slices they can access your dataset. So this is the workflow that how data submission is done and how it is managed on the main server. Now the sixth repository is Panorama. So Panorama is for the targeted proteomic knowledge base. So it is for the targeted proteomics, such as SRM or MRM. So it is an open source web application, and what it does is it integrates the Skyline files. So in the Skyline, if you have the targeted data set, you have analyzed it. So you can in the Panorama, you can simply open that Skyline file and it enables labs to organize and securely share both published and unpublished data. So similarly, in this case also, for example, I'm working on some mass spectrometry dataset and I have performed its analysis using Skyline. So what I can do is I can directly upload the files after the result files from the Skyline to the Panorama and I can provide access to different people from with whom I want to share my dataset. So you can share both the published and unpublished data through web interface so that you can support collaborations. And then it captures the complete data content from Skyline documents. So this is dedicated for the files which are coming from Skyline. So Panorama is like a knowledge base or a place like a library where you can provide your data, analyzed data coming from Skyline. So this is the Skyline. You have the data, you acquire the data, you build some method, you evaluate the results, you refine them. So this is the complete cyclic process that how you perform the data analysis in Skyline. So when you have analyzed your data, you have generated some results. So then you will submit it to Panorama, which will store it in a form of relational database, and then you can explore the results in a web browser. So what it does is that instead of looking it on your local computer on the Skyline app, you can upload your results to Panorama and anybody can access all those files, all those results or all those heat maps or all plots which you have generated. They can export them in the form of Excel sheets or plots, or they can use R programming to check more details. So this is all about the Panorama, which integrates the data from Skyline. This is one such example that human plasma.zip. So it is a file which is generated in Skyline. You can simply upload that zip file, which have multiple small files. So this is a zipped file and it contains 37 proteins and precursor count is 153, peptide count is 82, transition count is 459. This is a brief summary of the document or the file. And then in the precursor list, it provides a list of peptides and precursor note annotations, labels, Q1Z, Q1MZ. So this is the all the information related to all those peptides and their spectral informations. And then chromatograms. So this is the information of chromatograms, that in chromatography column, how the what is the retention time and what is the peak intensity. So this is plotted in this way so that you can visually interpret your data. So this is coming from Skyline. So in Skyline, different methods you have used and different analysis you have performed. Other than chromatograms, you can also have the peak areas, peak area and conditions, disease and healthy. So in this way, you can provide your analyzed data set to the means analyzed Skyline data set to Panorama, so that means all these results will be present. So anybody who want to see any information related to this analysis which you have performed, they can simply go through it. So this is one such helpful platform to share your data. Now we will look for different approaches in mass spectrometry because we want to look into the SWAT, that what is SWATH-MS. So before moving to that, we will look into few of the mass spectrometry approaches. So before moving further, I'll ask that till now, is everything clear? Okay, thank you. Yeah, so now we'll quickly go through mass spectrometry approaches. So first approach is shotgun proteomics. When you when you go for the analysis of entire spectra, you're not kind of doing the targeted spectrometry. You are looking for all the spectral information which is coming. So this is the high throughput analysis of complex protein mixtures by digesting protein into peptides. So in this case, you can identify thousands of proteins simultaneously, but you may miss low abundance proteins because there will be a biased sampling. So the proteins which are less abundant, they might not be captured. Then comes data dependent acquisition. So in this case, you select certain top and most abundant ions and you do not waste your time on all those known abundant ions. So in this case, this is the data dependent acquisition where you have decided a particular threshold yourself. So this is data dependent acquisition, and the strength is that it is high quality data for selected ions and it is well established. But yeah, the weakness is that because you are removing the low abundance proteins, so in this case, it is biased towards the high abundant proteins and it is of limited reproducibility. Then comes data independent acquisition. So in this case, you're not selecting top and abundant, but you fragment all the ions in specific mass range and collected the MSMS data for all the detectable ions. And it is unbiased because you are looking for all the spectral information which is available. You are not only selecting few ions and then letting it through MS2 spectra. So in the next slide, we will see that how DDA is different from DIA. Then comes targeted proteomics. So SRM, MRM. So in this, you are already aware that in this, we already have the information and we are particularly specific for a particular protein. So we already have the precursor to product ion transitions. And in this, we are only limited to specific smaller set of data. So we will not get information of other proteins or peptides which are present. But yeah, but we will be very sensitive and specific to a particular peptide for which we are searching. So that's why this is quite specific and sensitive. Then similarly, parallel reaction monitoring. So this is also similar to SRM, but it monitors all the fragment ions of a selected precursor. So in this case, in MS, after MS1, you select a precursor for which you want to pass it through the MS3, and means it will go for the further ionization or further fragmentation. So in the first step only, you select a precursor, and after that, all the fragment ions are captured. Then isobaric tagging, label free, and metabolic labeling. So this comes under the tagged mass spectrometry. So in the previous things, we are not tagging it, but when we are tagging it, so that is known as if we are tagging with the isobaric label, so that is isobaric tagging. If we are performing quantification without any label, so that is label free quantification, and which is quite cost effective. Similarly, metabolic labeling in the metabolic labeling we have select, so it incorporates stable isotope labeled amino acids into proteins. So these are some of the mass spectrometry approaches which are generally used for analyzing the data set. And these labeling are helpful to distinguish between different samples. So that's why this labeling is used, because when you are performing multi-reaction monitoring or parallel reaction monitoring, so in the same time, there are multiple data sets or multiple different samples. So in that case, labeling will provide you advantage to distinguish among them, and in a single run only you can perform your experiment. So now let's look at DDA and DIA. So in DDA, what we do is we have some threshold. So above that, we will select the only those ions or the precursor ions which have the intensity greater than a particular threshold. So these few of the labeled ions or precursors are only selected for the next round of mass spectrometric fragmentation. But in DIA, what we do is we divide it into smaller isolation windows, and then for that particular range of mass to charge ratio, we will perform the next round of fragmentation. So we will perform the fragmentation of all those precursors which are present here in each window. So in this case, fragmentation of selected precursors, only these colored precursors are selected after MS1. Whereas in this case, we have divided it into 5 different windows in this case. So we will fragment all these precursors in each window, and for each window, we will obtain the intensity to mass to charge ratio plot, and we will see. So in this, all the precursor ion which were present in a window, all information will be captured. Whereas in this case, only a single precursor ion was selected and it was undergone fragmentation. So in this way, it will have different uh, mass to charge and intensity plots for different precursor ions. And this is coming for different windows for all the precursor ions which were present. And if we look at the popularity, then previously data independent acquisition and SWAT were not that much popular, but with time now they are gaining popularity. And previously other workflows and such as shotgun mass spectrometric analysis and DDA were quite popular, but now DIA and SWAT are also gaining some popularity. So now we say what is SWAT-MS. So SWAT stands for Sequential Window Acquisition of All Theoretical Mass Spectra, and it is a data-independent acquisition method which is used in mass spectrometry for comprehensive and quantitative protein analysis. So what it does is that it divides my entire that plot of mass to charge ratio and intensity plot into different smaller windows. So there is some range which I decide, and that particular range will be the difference. For example, in this case, if I have a range of 400 to 1200 mass to charge, so I will divide it into smaller windows which will be of 25 dalton each, and we will rapidly scan all the fragment ions within that each interval in a single run. So in the next slide, I will show you that how it works. Now it achieves complete coverage because I will fragment all the precursor ions which are present in one single window, and it uses advanced mass spectrometers. Generally, triple time of flight mass spectrometers are used in SWAT-MS, and they are quite fast and precise for data collection. So most of the times we use triple time of flight. This is the most desirable or means best mass spectrometer which are generally preferred for the SWAT-MS, and it improves the quantitative accuracy and reproducibility also by directly constructing ion chromatograms from fragment ions, and it reduces issues like low repeatability, which is generally observed in the cases of traditional shotgun proteomics, and it is ideal for large-scale proteomic studies. SWAT means biomarker discovery and precision medicine applications, because it is looking into all the precursor ions. So in this case, we are not losing any information. That's why the SWAT-MS is most favored over the previous shotgun approaches. So there are different terms which you will encounter in SWAT-MS, such as window, cycle time, accumulation time, and Q1 isolation window. So these are the definitions of all of them. So window means that mass range is divided into smaller segments, which are called windows. So let's see in this figure. So in this case, uh, means this particular y-axis represents the mass to charge ratio. So I will divide these mass to charge ratios into different windows. So this particular arrow represents a single window. So this is a swath isolation window. So whenever you are looking into windows, you have to make sure that window will come on the mass to charge ratio side. So because in certain cases this axis might get changed. Some cases x-axis will become mass to charge ratio, and means it depends that how the people are representing the data. So in this case, the mass to charge represented in the y-axis. So this mass to charge ratio is divided into smaller chunks, which have the gap of 25 daltons, means 400 then 425. So they are separated by the 25 daltons. So this 400 to 425, it represents a single window, which is known as swath isolation window. In certain cases, I think Q1 isolation window is also another term which is referred to it. And then, so this is a window, which is the when the mass to charge ratio is divided into smaller fragments, that is referred to as window. And these each window will be of means equal size. It is not that a first window will be of 25 dalton and then next window of 35. So no, each and every window, its width will remain constant for entire range of the mass to charge ratios. Now the next thing which we have seen is cycle time. So cycle time, this specifies the cycle time. So uh, for example, in this case, for all these windows, whenever you are performing a single run for all the windows, so how much time is taken, that is referred to as cycle time. That is total time to complete one full scan of all the windows. So these are all the windows, and this they have not means they have missed few middle windows because there is a bigger gap from 500 to 1175. Otherwise, there will be similar arrows will also be present. So in the next slide, I have this. Yeah, so this is the mass to charge ratio, and these are all the windows. So the cycle time is this time. Whenever we see each window is separated from one another by some distance. So this distance is known as MSMS accumulation time, which means for how much time it was retained. So this is the accumulation time. So if I multiply my number of steps, so number of steps is nothing but it stands for number of Q1 windows. So number of windows is referred to as number of steps. When I multiply all these steps with my accumulation time, it will give me cycle time, that for each cycle, how much time was taken to complete the run for all the windows. That is cycle time, which will be calculated by multiplying number of steps, that means number of Q1 windows, by accumulation time. And this distance between the starting from the previous peak to the next peak, this time is known as accumulation time, and it tells you that this particular mass to charge ratio for how much time was it retained in the chromatogram. So this is the accumulation time. So cycle time will be proportional to the accumulation time. If more is the accumulation time, then your cycle time will also increase. Okay, so now next term which is present is the accumulation time. So yeah, we have seen that amount of time the mass spectrometer spends collecting the data for each individual window is referred to as accumulation time. Then Q1 isolation window, so it is also the same, and it is also one and the same thing. The isolation window, or the range of mass to charge ratio that the first quadrupole isolates for fragmentation during a scan. So if we talk about first quadrupole, then it is referred to as Q1 isolation window. And window width is the this particular width, that what is the range of that, what is the means what is the range between two different windows. So this is 25, so this is referred to as width. And Q1 isolation window also we have discussed, and accumulation time also we have discussed. So in this case, for each cycle or for each run, for each Q1 isolation window, we will have different means a mass to charge ratio to the time peaks. So this give me the intensity to the time peak. So in this case, I am running or I have five different cycles which are running one, second, third, fourth, fifth, sixth. So sorry, six different cycles. So in this case, we will eventually generate that number of means MS spectra means the number of times I am repeating the cycle. So in that case, that many number of plots will be generated for the spectra. And this is all about the SWAT MS plots, that how to analyze them. So these are few terms which were associated with the SWAT MS. So I hope it is clear. And then we have to balance the specificity with the sensitivity. So we have to determine that what should be my Q1 window size, because that will determine that what will be my accumulation time. So all those things we need to balance them. And total mass range can be calculated by multiplying Q1 width with the number of steps. So this is the Q1 width, which is the distance from here to here. So each window has some width. If I multiply this width with the number of Q1 windows, so number of steps is also the another term for the number of Q1 windows. So if I multiply these all steps by the width of this Q1 window, it will tell me the total mass range, which will be from here to here. So this is total mass range. And now we will look into the workflow of SWAT protein quantification. But before moving, I'll ask that is it clear? Okay, I will check. If some video will be there, then I will provide you the link, because that will be more helpful for you to analyze and remember these things. So now let's look into the SWAT protein quantification workflow. So first step is the sample extraction. Then you prepare the library. So for the library preparation, similar steps: enzymatic digestion is done, peptides are obtained, then LC-MS is done, in which you will generate the mass spectra. And then for the next step, you have to generate, after obtaining all those spectra, you will decide that which, because you have divided it into different windows. So in that case, you can decide yourself that which window you want to analyze or which particular cycle you want to analyze. So in that case, you can select them and you can perform the mapping them on the ion library. So similarly, for each protein, you will generate, for each condition, there will be the quantification for each peptides, because after getting the spectra, you will identify the peptide sequences to that particular spectra, and after identifying those sequences, you can decide that which peptides or which proteins you want to keep. So as we have seen in the data normalization or means in PC, all those analysis, we have seen that we just try to uh, reduce our data. We go for the dimensionality reduction and we make sure that we only keep the data which is mostly changing, so that if we want to perform comparative analysis, so that the proteins which are more variable, we can perform analysis for them instead of wasting our time on those proteins which are constant among both the conditions. So similarly, in this case also, we have selection for the target of interest. So in this case, particular proteins are selected for comparative analysis. And ultimately, for the target validation means you are selecting one particular protein which is means one particular protein which is more expressed in condition 2. So in that case, you can identify that particular protein, or you can also call them as target. For example, in cancer, if one protein is overexpressed, so that will act as a target or a biomarker for which you can design the drug, which is generally expressed in normal quantity in the one condition and overexpressed in another. So this is all about the SWAT protein quantification workflow, that how quantification is done. And this is just an example in which you have decided two different conditions and you are analyzing for those two conditions. And it depends on you, your workflow can change. That's why in each repository, they also allow you to upload the reanalysis results. So if you acquire new workflow, then your analysis will change eventually. So that is all about the workflow. And now in this slide, I've provided the advantages, that how the SWAT MS is better than the other already available mass spectrometry based technologies. So DIA is the data independent acquisition. So in SWAT, we go for the data independent acquisition, whereas shotgun proteomics, it comes under data dependent acquisition. And then comes selected reaction monitoring, which is very, very narrow, or you can say which is a targeted mass spectrometry analysis. So In each of them, if we look at the quantifiable proteins, so number of quantifiable proteins are more in case of data independent acquisition because we are observing for each and every individual precursor ion and we have divided it into multiple windows. So in this case, we will analyze more number of proteins, whereas in data dependent acquisition, I will miss out on some proteins because they were expressed in the lower quantities or their abundance was low. So in that case, I miss out for those proteins. And in selected reaction monitoring, I'm specific for certain peptides, so it will contain the protein information, very limited protein information it will contain. And if we talk about reproducibility, then SRM is most reproducible, then SWAT, and then shotgun. And similar for the sensitivity as well, because as these are targeted, so these are more sensitive than SWAT and then DDA. And this is the proteome pool coverage. So this has the maximum proteome pool coverage, whereas this has limited, and this is targeted, so this is very less. And similarly, dynamic quantification range and Mycobacterium tuberculosis ORFs. So this is just an example of this particular Mycobacterium tuberculosis. So how many theoretical ORFs and how many protein information is available in single run, how much information you can gather. So this is example of that. So ultimately they want to say that the SWAT is like a hidden part of iceberg. There much more information is present in SWAT as compared to other traditional or already currently running mass-based technologies. So in this case, they are telling you what are the advantages of using SWAT MS. And then these are some of the library-based DI, data independent acquisition software tools, which are based on the library information. So examples are Spectronote, OpenSWAT, Encyclopedia. So in this DIA, because Encyclop and DI is for the data independent acquisition, Scaffold DIA, Peaks Online, Peak View, Skyline. So these all are the tools in which you can go for the analysis of data independent acquisition, which is based on the spectral libraries or peptide libraries. So spectral libraries are they use the experimental data to provide you the precise matches for the detected peptides by comparing the fragmentation patterns, whereas peptide libraries they rely on the known peptide sequences without corresponding fragmentation data. So that's why these peptide libraries they are less precise than the spectral libraries, but they are more flexible in exploratory research. So these are the peptide libraries, and because in this you do not have fragmentation data, but you have generated those peptide libraries by yourself. Means what is expected. For example, if we go for the trypsin digestion or some digestion, then we know where it cleaves. So for example, if I have the information of a protein sequence, then I can simply generate the expected peptide spectra from there. So that peptide library is that way. And spectral library is that experimentally when you from mass to charge ratio determine the peptide sequence. So that is referred to as spectral libraries. So these are the softwares, and if you want to read in detail, then you can go through this slide. And similarly, if we talk about library-free DIA software tools, then those are MS Fragger DIA, MaxQuant, DIA NN, and DIA Empire. So these comes under library-free DIA software tools in which we do not use the already available library information. This is high-speed search engine for DIA data and part of the fragmentation pipe, which is the fragmentation pipeline, which is designed for library-free analysis. So in these scenarios, you go for the quantification of your mass spectral data set, and you do not require the library information to go for this particular analysis. So that comes under library-free DIA software tools. And then comes the tools which which can both be library-based as well as library-free, such as DIA NN. So this is the fast DIA analysis tool which uses neural networks, and it supports both library-free analysis along with the spectral and peptide library workflows, which is library based. And another one is DISCO, which stands for Data Independent Acquisition Signal Correction. So this is also open source tool which supports both library based and library free DIA analysis, and it focuses on accuracy and reproducibility. So hybrid DIA software tools are favored over the already available library free and library based. If you want to go for the hybridized analysis, because it is based on both those analysis, so it will be uh, more accurate and more reproducible, and more information will be generated by them. So this is just for the your sake of understanding, so that because there was one question related to these tools, so that's why I covered them. Now you are supplying your data to repositories, so there are some kind of standards which have to be maintained. So which comes under the quality control, that what kind of data you are submitting. So there are some assessments which are done on the library, so which refer to as DIA library QC. So this tool allows the users to analyze the DIA libraries in variety of formats, including PeakView, OpenSWAT, SpectroNote, PredictedIA libraries based on library characteristics. So in this case, you can assess the libraries which are coming from data independent acquisition. So all that assessment can be done using this particular tool, and it can check for the complexity, characteristics, modifications, completeness, correctness. So these are few parameters which are analyzed for the already available library. So you can decide that which library you want to choose and what are different things. So all those things you can check using DIA library quality control. And this is the workflow. Assess the spectral library. For example, you have the SWAT definition file and proteome mapping file. So by using DIA library quality control, you also have an ion library. So you can analyze the library, that whether it is correct, whether it is complete. You can generate a plot. So this comes under assessing the spectral library. And you can also correct the spectral library. So in that case, you can inspect the diagnostic criteria. You can check for the mass error. For example, if there is some mass to charge ratio and the peptide which is generated, there is some error in the mass. Means, for example, if one amino acid sequence is given and that particular amino acid sequence does not correspond to that particular mass to charge ratio. So in that case, you can check that is there any mass error. If there is, then you have to use the DIA library QC. You have to recompute the theoretical mass and you have to correct that mass in the library. If no, then you will look for the conflict assays, whether there are some conflict in the assays or the analysis which have been performed. So you can use DIA library QC and you can filter the problem assays. You can either clean the library or you can see what is the problem which is present in the library. And if there are no conflict assays, then you can simply use that library for DIA analysis. So DIA library quality control is just to make sure that the library which is present in the DIA, which is supplied by DIA, is whether it's accurate or is there some issues. So this is to means increase the correctness of that library and to decide that whether that library could be used or not. This can be done using DIA QC. Then there are MS-based repositories. So these three repositories which were majorly discussed, SWATHAtlas, SRMAtlas, and PeptideAtlas. So this table tells you the entire information about them. But the major difference between them is the data, the experiment which are performed. So in SWATHAtlas, it will be entirely related to the DIA-based mass spectrometry data. SRMAtlas will be entirely about the targeted mass spectrometry data. And PeptideAtlas is like a comprehensive collection. So this is not specific to SRM, MRM, and this will contain the comprehensive all the collection of peptide identifications, all types of mass spectrometry experiments. And then if you want to read in more detail, then you can read this. This is just for your understanding. If you want to go through in detail, then you can go through this slide. Yeah, so now I will talk about the collaborations, that what are different collaborations related to at the level of genes and at the level of proteome, that how different resources have been collaborated among one another, just like ProteomeXchange. So ProteomeXchange is a consortium which is specific for mass spectrometry data. Similarly, there are other different consortiums which are for proteome dataset, not just mass spectrometry, but all the information related to proteins which are involved in different conditions. So before moving to that, I will just check that is there any questions related to the previous content which you discussed? Okay, now let's see. So we have 20 minutes. Okay, so now there is an Office of Cancer Clinical Proteomics Research. So just like ProteomeXchange, which is for all kind of mass spectrometry data, similarly there are some other consortiums which are quite specific for one particular condition. So similarly, this particular office, this is specific for the tumor categorization programs, which will provide you the data which is specific to cancer. So in this case, the different repositories are Clinical Proteomic Tumor Analysis Consortium, Applied Proteogenomic Organizational Learning and Outcomes, and International Cancer Proteogenome Consortium. So these consortiums, they already contain information from multiple repositories. So these three consortiums come under tumor characterization programs, and they keep information related to cancer specific proteomic information. So where is the data? And if we talk about genomic data, then it is available in NCBI in the form of SRA, which is Sequence Resource Archive, and Genomic Data Commons. So there are two kinds of data. One is proteomics data, one is genomics data. So similarly, if we talk about genomics data, then it is available in Genomic Data Commons, in which multiple genomic dataset repositories, they combine together and they share their data at one single place, which is known as GDC, which is Genomic Data Commons, which is a single place from where we can access data related to, means genome data related to different cancer specific genome, and it is hosted at NCI. So it contains clinical data information as well. Similarly, CPTAG, which is for the proteomic based data, proteogenomic data. So this comes under Proteomic Data Commons, which will keep all the data which is specific to cancer, but it is for the proteome level. So this is the proteomic data. And then there is also additional imaging data, so which is stored in the Cancer Imaging Archive, which will tell you about the different images, so staining experiments and all those experiments. So in that case, when imaging data is generated, it will be in the Cancer Image Archive. So this all data is specific to cancer. So in this case, the proteomic data will only be specific to the cancer. Now, Clinical Proteomic Tumor Analysis Consortium, which is CPTAC. So we have also previously discussed about the CPTAC. So its builds are on TCGA, which is tumor characterization, which comes under tumor characterization program. And this contains information specific to the genes which are expressed in the tumor condition. So other than this, it also have the resources such as genomic data, proteomic data, imaging data, fit for purpose assays and antibodies. So these all things are available in CPTAG. And this is the CPTAG pipeline, that how it works. So from where it collects the data, tissue sources atlas, tissue sample and clinical data is taken, then biospecimen core resource, which will perform pathology and molecular quality control, which will check that whether these particular information, the information coming from tissues and clinical data, whether those proteins are related to some kind of pathology. So those analysis and quality control analysis will be done here. Then protein, DNA, and RNA will be obtained. Then they will be characterized in proteome characterization centers. Then primary genomic and proteomic data will be obtained, which will be specific to those data types, such as whether they're involved in some kind of disease. Then fourth step will be the primary and uniform genomic and proteomic data, which will be provided here in the proteogenomic data analysis centers. They will analyze the data, they will analyze the results, and then CPTech data coordinating center will be there, which will provide that data for further analysis. So primary and uniform genomic and proteomic data, it will be further analyzed. Similarly, here also the analysis will be done, which comes under proteogenomic translational research centers, which will perform the analysis, and they will also check that whether what are the drug responses and whether there is some resistance in certain proteins or in certain samples, or in, there are some people who are resistant to certain drugs. So all those informations will be generated in proteogenomic translational research centers, and ultimately this entire information will be provided to the research community. So this is a CPTech pipeline, beginning with the information coming from the samples to core analysis, and ultimately providing that information to the research community after different centers are present, which analyze the data and add on more and more details to that data. Other than protein information, it also adds the information such as drugs or pathological metadata. So all those informations. Then comes GDC, which is Genomic Data Commons. So you have previously seen that how it organized the information from different projects, and you can explore them. You can check for different projects. You can perform analysis from the repository. You can see which proteome information is available. So there are different tissues for which the information is available. So these are the tissues. Most of the information is for the lung tissue. So in this case, you can click on any of these bars, and then it will take you to that particular tissue specific data set, which will be at the level of gene level data. So in genomic data common, it will be a gene level data. And this is a summary that what are different genes which are present, what are the mutations. So in cancer, if one particular gene is expressed in a particular tissue, then in cancer, how it changes, what are the mutations, what kind of mutation is there. So all that information you can obtain from the GDC. And if you want to read further about GDC, then you can see it here. It tells you about entire information about GDC, who have developed it, what was the aim, and all those things you can read in the slide. And then this is the main purpose, that why the genomic data common was developed and how it brings about the entire information at one single place. So this is just like a small workflow, that it is a knowledge base for cancer that promotes sharing of genomic and clinical data between researchers and facilitate precision medicine. So that's why genomic data common was developed, so that in single place only people can access the data and they do not have to go through multiple repositories. This is also a single point access through to multiple repositories, and it brings together the harmonized genomic datasets. So it contains a large information, and it integrates genomic and clinical data, because clinical experiments are also running side by side, and it integrates both of them, and it provides the data such that people can export that data and they can perform analysis to it. So in that way, it expands the access to genomic and clinical data. So this just tells you about the purpose, that why genomic data common was developed and how it helps to provide the data to the researchers. And then GDC data submission metadata are as important as molecular data. So yeah, because other than raw data, metadata is also important, because it will tell you that how your data is linked to one another. So in this case, when you integrate your data, which is already available, so it becomes the metadata. So in GDC, it also provides you the metadata. This is example of the biospecimen data, and this is the example of the clinical data, and this is the example of a molecular data, that how different data files, what information those data files keep and how they are linked. So that information is also relevant, which will be provided by GDC data commons, which tells you the relationship between the elements described in GDC data model, because there are different information, such as, for example, if you talk about any particular case. So for example, breast cancer, so it will tell you about the demographic, in which regions it is mostly observed, what are the diagnostics, what are the exposures, family history, follow-ups, molecular test treatments. So all those information to one particular case will be given. So this is known as metadata, when entire bigger information is provided. So all that information you can find in GDC. And this is further about the different, means it provides you the access to repository exploration and analysis. So this is a bit detailed about that. Similarly, this is the home page, which we have seen in the previous lectures also. And this is the slide about the proteomic data common. So similar to GDC, which is the genomic data common, proteomic data common is also similar initiative, which is to bring all the proteomic information on the one single platform. And these are the benefits, that means it brings it to a smaller place so that anybody can access it. It can be searched on the cloud-based infrastructure. Analysis can be done on the cloud. So this is the advantage of bringing the entire data to a smaller common point. And this revolutionized the precision medicine, because you can easily share the data. So from about, you can read about this. If you explore it, then it will tell you about different data which is present in this particular proteomics data commons. So different sites for which the proteomic data is present. So in that case, you can select the primary sites, different programs. So in proteomic data common also, there are different programs which contribute the data. So different programs are taking information from different means repositories. So these programs, they all combine together, they share their data. So you can also decide any such program. If you want to be specific to one such program, then you can select it from these checkboxes. And ultimately it will provide you the result in this form. This tells you analytical fractions. So acetylome, like glycoproteome, lipidome. So this is just the example of all the analytical fractions and disease types, which are majorly listed, and the experimental types, that what kind of mass spectrometry experiments were done for the proteomic analysis and how many cases are present. So this is similar like the statistics related to this particular proteomics data commons. And it now in this table, they provide you about the studies which are present, biospecimens, clinical analysis which is present, files, genes. So all those informations you can access from here. In the table, you will find the ID, which is specific to the proteomic data common ID. Then what kind of study? So in this case, it is taken from the CP tag. And what was the project? What is the program disease type? So this is specific to glioblastoma, because we have seen that GDC and PDC, they are specific for the cancer specific data, cancer specific proteomics and cancer specific genomic data. So they will be specific for a disease type, and most probably these diseases are from the human. So the species will only be human, whereas this was not the case in ProteomeXchange. So this thing you have to keep in mind. And then available files. So it will provide you raw files, process mass spectra files, metadata files, PSMs, and protein assembly. So it depends, because each program have certain files. Some might not provide you with the metadata files, but some can provide you. So in that case, you will find some numbers. So this is about the proteomic data common, and this is the common pipeline. If you want to go through this, then you can read it, that how the information is arranged. PDC object store, then raw data, and then it is converted to MZML and MGF file. Then there would be a localization score and peptide spectrum matches. Similarly, database search. So you can simply go to localization score, or you can simply search the database. After obtaining the mass to charge ratio, you will find the peptide sequence, and then you will check for the quantitative analysis. You can quantify that peptide expression, and you will identify the gene or site level reports. Or you can also perform the MS1 data processing, integrated MS1 peak areas. So this is about the means how the DDA based analysis is done on the proteomic data common, because after obtaining the mass spectra files, you can decide. So in this case, this is a data dependent acquisition. So after the MS1, you select specific mass to charge ratio peptides and then proceed with that. This is a simple pipeline, that how it is performing tasks. In analysis, it tells you about explore quantitation data, peptide genome mapping, common data analysis pipeline. So this is common data analysis pipeline, which is coming from this particular menu. Similarly, you can explore others also if you want to read in more in detail. Yeah, and this is also one such data submission and release policies. So whenever you submit your data, then there are certain policies which you have to follow. So if you want to read it in detail, then you can go through this slide. But as we have already discussed, that what happens, that you have to register with the PDC workspace, then you have to upload your raw data, you have to create a project which will contain that data, because your project can contain multiple data files. So in such cases, you have to create a project. Then you will create a protocol, that how you have designed a workflow. So in this case, you will enter a workflow, that stepwise procedure, and then you will create a study. So this is about the how the data is submitted in the proteomic data common, and these are the policies which you can read. And then this slides tell you that what was the vision, that why the Cancer Genomic Data Center was created. So these are the uses, that it identify low frequency cancer drivers, define genomic determinants of treatment response, compose clinical trial cohorts, sharing targetable genetic lessons. So this paper is specific for the means why the data genome means genomic data common was developed, and what advantages it provide, because everything is kept at a same place, at a single place, single point access is provided. So how it is helpful, and it has helped a lot in the cancer specific studies, and now they are trying more and more so that better personalized medicine can be developed. So this paper is for that. If you want to read in detail, then you can click on this link and you can go through this paper. And then there was another review which was about the importance and challenges, possible solutions for sharing proteomic data while safeguarding individuals' privacy. So this paper was also nice, in which they are telling you that if you are uploading your data, then you do not have to worry. There are some guidelines which are already available, through which you can share your data also, and you can make your data private. So your data won't be stolen by anyone. So this is related to that particular data, means that particular concept. So you can read this paper if you want to. And then there are some guiding principles which should be means followed for your whenever you are submitting your data. Then there are some guidelines which you need to meet for submitting, which are known as FAIR guiding principles, which are required for scientific data management and stewardship. So These are FAIR, which stands for findable, accessible, interoperable, and reusable. So what does these mean? Findable means that whenever you are providing your data, so it should be findable. Anybody should find your data. Machine should be able to find it. Human should be able to find it. So how they can find it? Because there will be some kind of identifier which will be linked to your data. So it should be findable. Then comes accessible. So that means the data which you are providing, it should be accessible. It should be people could able to download it, or they used to means they can be able to access it. So that comes under accessible. Then interoperable. That means if some person is downloading your data, so even if he exchanges machines, it should be you should be he should be able to work with that data on different machines. He should not be restricted. So that comes under interoperable. That means you should use the standardized terms such as controlled vocabularies so that everybody could be able to access it, and machine should also be able to action on that. So that is known as interoperable. And then reusable, which means your data should be well described for both human and computers to understand it, and clear and accessible data usage license so that anybody could reuse your data and perform the reanalysis. So these things must be fulfilled while submitting your data, and these are the properties which your scientific data must have. And these are the stages. So first stage is big data source, in which you have unstructured, structured, or proprietary data. So this is the data which you have. Second stage is security, cleansing, and standardization, because in FAIR principles, you have to make sure that everybody should be able to, it should be interoperable and accessible. So for that case, you have to clean it, you have to perform data transformations, you have to standardize your data. So clean and score. Then comes stage three, which is analytics and quality control. So it will check for the data whether the things which are arranged they are correct or not. So analytical process and quality will be checked. And then comes the deliverable step. That means now your data which you are submitting, it is completely checked, it is confidential, and it is correct. So now means after deciding how means to whom you want to provide the access, ultimate step is the deliverable, where you will provide the reports or the dashboards, which is the final delivery, which means that you have submitted your data and now it is delivered to the public and they will be able to access it. Similarly, there are some proteomics standard initiative which are for the proteomics dataset. So there are some standards which are designed by the Human Proteome Organization, which are referred to as Proteomics Standard Initiative. So these generally decide that globally how to represent the data so that anybody sitting anywhere can understand that. So it brings the data to a common platform. And these are the work groups which comes under proteomics standard initiatives, such as molecular interaction, mass spectrometry, proteomics, informatics, protein modifications, protein separation. So they decide the vocabularies and minimum information specification. So minimum information specification means that with the minimum of the minimum information, you should be able to uh, communicate your data. So minimum information specification means that you have to write a minimum information, but it should be sufficient to describe your entire data. So that is known as minimum information specification. And these decide the vocabularies and support for implementation of standard in the public available tools. So this is just to standardize your data so that there are not any problems, so that it should be on a common ground and anybody can understand it easily and anybody can access it. Similar to the FAIR guidelines, HUPO is for the proteomics, that it manages that your data is properly organized and it is standardized. And under HUPO, you will also find the Human Proteome Project, which was specific to understanding the human proteome, such as the Human Genome Project. Similarly, Human Proteome Project was also established, and which was established by the HUPO. So if you want to read about it, you can simply read about this. What is the mission of this, and who are the people who are working for the Human Proteome Project. And similarly, they have mentioned about the 4 pillars of the Human Proteome Project. So these 4 pillars are mass spectrometry, antibodies, knowledge base, and pathology. So these are the 4 pillars from where this proteomic data is coming, and these form the foundation from where the Human Proteome Project is taking the data. Then chromosome-based Human Proteome Project and biology or disease Human Proteome Project. So protein information are also rendered from the disease-specific samples. So similarly, all those informations generate a human proteome project, because each and every day new peptides are coming. So in that case, new proteins are also identified from those peptides and from the sequencing methods also. So this comes under the human proteome project. Now we will see a few MCQs and then I'll end the session. So till now, is everything clear? Because I might have rushed through the slides in the end. So, okay, now let's see a few MCQs. So you have to match the following proteomic standard formats with the steps from which they generate. So these are the standard formats and these are the steps. So you have to tell that which step will generate this particular format. So the first one is MZML, second is MZIdentML, third is TraML, and fourth is MZTab. So you have to tell which format is generated from these steps. Okay, so now do you have any idea? Because certain things are quite easy, which you can simply mark, which you can simply identify that to which that particular format will belong. So can any one of you guess? Okay, so let's see. So the correct option is option D. So MCML, it is for the MS data because mass to charge ratio, and then MZIdentML, so it is for the identification data. So after identification, the format will be MZIdentML. Then comes TraML, so this is for the SRM, and MZTab is for the final results. So these are the formats for these steps. Now let's look for the second question, which is the Sushant is planning for the SWATH-MS experiment. For best results, which mass spectrometer you would suggest him for his experiment? So option A is triple time of flight mass spectrometer. Option B is triple quadrupole mass spectrometer. Option C is mass spectrometer with Orbitrap mass analyzer. Option D is mass spectrometer with ion trap mass analyzer. And option E is mass spectrometer with Q executive. Okay. What about Ptolemy? What do you think? Which mass analyzer should he use? Which mass spectrometer he should use? Okay, so you are saying A and E. So correct answer is option A, which is triple time of flight mass spectrometer. So triple time of flight is mostly, it is suggested because it is for the best results. So that's why triple time of flight is mostly preferred and not the Q executive. Now the third question is, which is the last question, which of the following mass spectrometry experiments could provide highest depth over coverage in a proteomic experiment? So shotgun discovery experiment, shotgun experiment with inclusion list, selected reaction monitoring, and SWATH-MS experiment. So highest depth and coverage, which means with which you can identify more number of proteins and more number of peptides are covered. Okay, so both of you have said option D. So yeah, it's correct. And we have also seen in that slide where we have compared among SWAT MS, shotgun, and selective reaction monitoring that how the SWAT MS has improved the depth as well as coverage in experiments. Okay, so this was all about the today's session. So do you have any queries, or should I end the session? Okay, so last time I have searched for that book, but I could not find the PDF which is freely available because they are saying that for the ebook you have to pay some amount. But I got the kind of a sample pages for that particular book, so which I have uploaded in the same drive. So these are some of the sample chapters, and I think they cover some of the topics which are given in these proteogenomics means weeks. So you can go through that model chapters also. That will also provide you some kind of help. But I was not able to access the entire ebook. Yeah, although I tried on other means online servers also from where you can download books for free, but this book was not available. So, but I hope you can go through that particular model or the sample pages. So that will also provide you more of information because I looked into that and I checked that it was having the sufficient amount of information. So that will still help you. Okay, then I will end the session. Thank you so much. And I will meet you on the next week.